Welcome everyone to the Neurons Matter seminar. And it is our pleasure today to have Dr. Siddhartha Sikdar. Dr. Sikdar is currently a professor in the Department of Bioengineering at George Mason University in Virginia. He's the director of the Center for Adaptive Systems of Brain-Body Interactions, whose faculty and students pursue transdisciplinary research to enable all individuals to fully participate in needed and desired life roles and activities. Dr. Sikdar's research group conducts translational research using imaging to investigate brain-body interactions in a number of clinical conditions of major public health significance, such as chronic pain, stroke, spinal cord injury, and amputation. His group uses state-of-the-art ultrasound and laser instrumentation for developing new ultrasound, optical, and hybrid imaging technologies and assistive technologies. He's the recipient of the NSF Career Award, numerous awards at his university, and his research has been funded by the major agencies, National Science Foundation, National Institutes of Health, Department of Defense, and he is a principal investigator on an NIH HEAL initiative to develop imaging biomarkers for myofascial pain. We welcome you today, and please take it away, Dr. Sikdar.
Thank you so much for that very nice introduction, Greg, and thank you for inviting me to speak at your seminar series. I'm very excited to be here, and I have been looking a little bit about the work that you are doing at your center, and it's really exciting, and I think I'm hoping to have a good discussion at the end about some of the work that we have been doing and how that might potentially overlap with your interests. So as Greg mentioned, my background is primarily in imaging. I'm a biomedical engineer, so my core methodological expertise is in imaging, but I have been working very closely with a number of faculty from many different disciplines over the last 15 years or so at Mason, and including physiatrists, neuroscientists, physical therapists, and other sort of data scientists and engineers. And we have been trying to grapple with a number of different problems. And one of the problems that I'm going to talk about today is chronic pain. So I also direct a transdisciplinary center at Mason, and the center sort of brings together faculty from all over the university. And this was one of the 5 transdisciplinary centers that Mason that started in 2018. And as was mentioned, the vision is to enable individuals with disabilities to participate in needed and desired life roles and activities. And our emphasis actually has not been to focus on any one particular condition, but also to look at a number of different conditions that are leading causes of disability and death. And in particular, because I think a lot of groups have developed a lot of deep expertise in particular conditions, but often the cross-pollination between best practices in one population are not always applied in other populations. And the other thing that we recognize in our center is that many of these are often co-occurring. So for example, there is a strong overlap between substance use and chronic pain because these are sort of ancient mechanistic pathways in our brain that are involved. There's also a lot of overlap between mental health and behavioral health disorders and chronic pain and substance use. We also study a lot of musculoskeletal injury and stroke, and there's also a lot of co-occurring overlap between multiple conditions. So in our center, we have therefore decided to not focus on any one particular pathology, but try to see what are some of the commonalities and differences and best practices that we might be able to learn from each one of these conditions. So that's sort of a very brief background about our center. So our methodological expertise really is in multi-scale imaging and sensing. So we have a lot of different faculty who are involved in looking at both tissues, but also functional and physiological measures at different scales. We also have a group that is working on neuromodulation using transcranial magnetic stimulation, transcranial DC current stimulation, some using focused ultrasound. So there's a lot of work going on in neuromodulation. We have a deep expertise in human movement analysis. So we have a number of faculty from kinesiology and biomedical engineering who are interested in human movement, multiple scales. And some of our imaging work is actually closely tied to this human movement analysis. We are actually developing technology that allows us to use a wearable ultrasound to dynamically look at muscle activity while individuals are moving. We also have a number of faculty who are working on assistive technologies. My group, for example, has been developing new ways of controlling these advanced prosthetic devices. We have some faculty who are working on exoskeletons for stroke rehabilitation. And again, sensing and imaging are part of those efforts because we are trying to understand volitional intent and try to recognize volitional intent using these different sensing modalities. And then there is a group of faculty who are involved in behavioral health, and they're using virtual reality and other technologies to look at behavioral health. So it's a very broad center, but it's also very exciting because we tend to learn a lot from each other. Today I'm going to talk about our work on our chronic pain team, and in particular, we have been working for almost more than a decade on myofascial pain. And myofascial pain, as many of you probably know, has been thought of as a regional pain syndrome for many years. And it is primarily a non-articular pain that affects soft tissues. But increasingly, we have recognized that myofascial pain is pretty ubiquitous. It is not only just an isolated regional pain syndrome, but also co-occurring with pretty much all the other chronic pain syndromes that we are aware of. So we are very interested in trying to understand, what might be going on at the level of soft tissue in all these different chronic pain conditions. So our group, again, has brought together imaging folks, data science, clinical science, neuroscience, and as well as a number of different physicians who treat patients with different interventions to try to make sense of all of this. So Greg mentioned the HEAL initiative, and I am sure most of you have heard about the HEAL initiative. So this was initially started with a focus on addiction and opioid, considering treatments for opioid use disorders, but there's been a lot of effort more recently on trying to figure out non-opioid treatments for chronic pain. And a couple of years ago, this new initiative was launched to try to understand myofascial pain as a core element of the chronic pain research, because this is one of the least understood aspects of chronic pain. So the motivation is that most of the current treatments for musculoskeletal pain are inadequate. For example, just to give one example of low back pain, it has been reported that 95% of low back pain could potentially be non-specific or essentially undiagnosed or unexplained. And so the diagnostic algorithms for identifying red flags that are treatable only impact a small portion of the population. And then the rest of them really, there's not a lot of evidence-based practices for treating the underlying mechanistic pathways. So, and part of that is because we do not really understand how the pain perceived in these soft tissues are involved in these chronic pain conditions. And we also primarily rely on palpation right now for diagnostic criteria, and they're highly subjective. And it's become a huge challenge for doing any kind of research because the gold standard criteria are subjective and controversial. So it becomes very difficult to develop quantitative tools when it's not entirely clear what is it that we should be looking for. So that's part of the the effort in the HEAL Initiative biomarker program is to develop more quantitative methods to understand the physiology and function in these soft tissues. So today I'm going to start with talking about what we call the myofascial unit. And this is an evolution of our thinking from thinking of myofascial trigger points, which are ubiquitous in the literature. That's often a palpable physical finding that is considered to be a hallmark of myofascial pain syndrome. But we have increasingly realized that focusing just on the trigger point is perhaps missing sort of the bigger picture of what might be going on in the soft tissue. So we have moved away from just thinking about trigger points and thinking a little bit more comprehensively about what might be going on in this myofascial unit. And I'll talk about what I mean by that. And then I'll talk a little bit about some of our thinking on the mechanistic basis and the evidence for that. And then I'll just present some of our data on the imaging evidence, and then we'll talk about some opportunities and limitations. So what exactly do we mean by a myofascial unit? So one of the recent pieces of evidence that has emerged is that the fascia, which is the loose connective tissue and that's sort of this hierarchical organization of connective tissue that is ubiquitous throughout the body, is actually very interesting. So it used to be thought of as a purely passive sort of the structural tissue, but actually it has become obvious that there is a lot of interesting physiology that is happening in the fascia. First of all, the fascia is highly innervated as one of the most densely innervated tissues. There is a lot of the sort of the proprioceptive architecture is localized in the fascia. So the muscle spindle's capsule is actually continuous with the perimysial connective tissue. They are richly innervated with neurons that are nociceptors, as well as free nerve endings. And also the vascular apparatus is obviously sort of localized in the fascia, including the lymphatic pumps that regulate the fluid exchange and so forth. So there's a lot of recognition about the role of fascia in force transmission from the muscles to the joints. But there's also interesting sort of neuroimmune mechanisms that might be going on in the fascia that may play a role. So our thinking is to not think of muscle fibers in isolation and to study a muscle, but also to look at this entire sort of functional and anatomical complex that includes the fascia and the connective tissue, the innervation of that, the blood vessels that supply it, as well as potentially the lymphatic pumps and lymphatic vessels as well. So thinking of this entire sort of functional and anatomical unit in as sort of a unit of analysis and imaging it could potentially lead to new insights. So that is sort of what we mean by the myofascial unit. And so this is a little bit of a generalization from what is reported in the literature. In the literature, often people will only think of the myofibers and the connective tissue as the myofascial unit, but we are including nerves, blood vessels, lymphatics, as well as part of this complex. So here are some key observations that sort of are the basis for a lot of our thinking on this problem. So the first observation is that in chronic pain, of course, sensitization in the dorsal horn is one of the hallmarks. The term central sensitization is often used, but central sensitization can often be vague in some ways. So in our work, we are not necessarily talking about sensitization in the brain, but we are specifically talking about sensitization in the dorsal horn, and that trying to understand how that might be related to dysfunction of the myofascial unit. Of course, psychosocial factors are a key part of chronic myofascial pain and musculoskeletal pain in general. And the question is, how are these psychosocial factors playing a role? So that's an important observation that we need to be able to explain. The other observation is that the soft tissue dysfunction is coexistent with many other underlying conditions, including visceral conditions. So, for example, irritable bowel syndrome, endometriosis, all of these have been associated with myofascial pain. And so the question is, what is the link between the soft tissue dysfunction and potentially visceral mechanisms? And then a number of different types of abnormalities have been reported at the myofascial unit. There have been some structural abnormalities reported, mechanical, neuroimmune abnormalities. So how can we try to better understand what might be going on? So these are some of the motivations for what we are trying to answer. So the first key question is, what is the mechanistic link between sensitization in the dorsal horn and dysfunction in the myofascial unit? So that's a question that needs to be answered. And then what is the link between the dysfunction in the soft tissues and the pain perception itself? So that link itself is also not fully elucidated. So let me start by sort of summarizing the reported sort of domains of dysfunction in this myofascial unit. So of course, there is a lot of literature on sort of changes in the structural, the structure of the myofascial unit, including there have been changes in the thickness of the fascia. There's been some evidence that there might be some micro tears and scarring or fibrosis going on in that in the milieu. There's been some reports of disorganized architecture, although these have not been fully confirmed in large studies. We know that in some cases, there might be fatty infiltration as part of the alterations in the muscle and the connective tissue. But again, it's unclear what the role of that is in myofascial pain. And of course, the primary findings of these trigger points and taut bands in myofascial pain is mechanical findings. So it's confirmed by physical exam, but now increasingly being confirmed with imaging and other methods as well. So we know that there is regions of increased stiffness that are palpable. With other methods, there's been reports of reduced shear strain, shear plane mobility alterations. So there is some evidence of the thickening and the densification of the connective tissue, not only the epimysial tissue, but also the perimysial and intramuscular fascia that appear to be densified and thickened and altered in terms of their mechanical properties. And then there have been reports of sort of vascular and immune and metabolic findings as well. There's been data that show that there is an alteration in the local inflammatory milieu. There are some evidence of reduced pH, some evidence of chronic inflammation that is somehow not being cleared. And there could be potentially dysfunction in the lymphatic system that's failing to clear the local inflammation. And then there are other sort of isolated reports of some metabolic dysfunction as well. And in our group, we have shown that there are some changes in the blood flow in the local milieu. So these are the sort of spectrum of findings that have been reported in the literature. So our group has developed a sort of a hypothetical framework for what we think might be going on. And it is important to point out that the evidence is still not comprehensive. So there are some evidence for pieces of this puzzle, but it's not been elucidated in a complete circuit yet. But what we think might be going on is that there could be a primary source of nociceptive bombardment that may or may not reside in the muscle. It could be in the muscle, but it may be elsewhere. So for example, there could be a visceral source of nociceptive bombardment. So endometriosis could be the source. It could be irritable bowel syndrome could be the source, or some other underlying source. It could also be in the joint. So for example, in low back pain, it could be a facet joint dysfunction. It could be in the disc. So there could be a number of underlying causes that leads to this bombardment in the dorsal horn, and that causes sensitization. That means the lowering of the thresholds and sort of the plastic changes in the spinal cord. And this part is fairly well understood. So the changes in the spinal cord as a result of nociceptive bombardment, that those pathways have been well elucidated. The next link is to understand what might be happening in the muscle as a result of the sensitization in the spinal cord. And this is where one of our colleagues, John Strobel, has published a series of papers that show that there is some evidence of neurogenic inflammation. So in other words, antidromic mechanisms that cause local inflammation in the milieu of the myofascial unit as a result of sensitization in the spinal cord. So these are essentially neuroactive substances that are being activated in the local milieu through these antidromic mechanisms. And so there's some evidence for this. This is not fully understood, but there are increasingly more and more evidence that there could be local CGRP, substance P, etc., in the local milieu of the muscle as a result of sensitization caused in the spinal cord. So if this is indeed true, then a lot of the findings in the myofascial unit can start to make sense. So if there is a local change because of inflammatory mechanisms, then there are some mechanisms that have been proposed that can lead to alterations in the fascia itself. And so the aggregation of hyaluronan could, and then sort of myofibroblast activation in the fascia could lead to this kind of observed thickening of the fascia. And that could also have these kind of vicious cycle of downstream effects, including impairment of lymphatic drainage, alterations in the blood flow, as well as sort of this the thickening of the fascia could alter the proprioceptive mechanism. So there could be this sensory motor mismatch happening in the fascia as well. So this in turn could become a vicious cycle that perpetuates this perception of pain. So the hypothesis is that in a number of these pain conditions, there could be a soft tissue component that is involved and is critical in sort of maintaining this kind of pain perception. Because there is this link between sensitization in the spinal cord and the local changes in the myofascial unit, we can then hypothesize that the pain neurometrics could be modulating this as well. So there could be descending mechanisms that could be modulating the sensitization in the spinal cord. And so that provides a sort of a plausible link between how psychosocial factors might be affecting sort of perception in the soft tissues. Again, these are hypothetical mechanisms at this point, but this is a framework that can explain a lot of the observations that we know to be true from a clinical perspective. So our group has been trying to develop some of the data to try to see what might be, whether what we see is going on in the local tissue is consistent with this framework. So the first thing about this, the neurogenic inflammation. So as I mentioned, Dr. Strobel has published a number of papers that show that if you, in an animal model, if you induce sensitization through some kind of injury to the spinal column, there is now this dorsal root reflex that leads to the release of pro-inflammatory neuropeptides like substance P and CGRP into the segmentally linked tissue. So there is some mechanistic evidence that this might be going on. And the question is, is that what we see in humans as well? And that is still being investigated. The other piece of evidence is that the fascia are altered in myofascial pain. And so our colleague Antonio Stecco and Carla Stecco have shown in very detailed studies of how these changes in the biomechanics of the fascia may play a role. So There's evidence that there's a densification of the hyaluronan in the fascia, and this densification impairs the gliding of the fascia. So in other words, the biomechanical force transmission through this myofascial chain is altered because of the alteration of the viscoelasticity, the connective tissue. And the cause of that is still to be elucidated, and it is possible that the local inflammation may be one of the mechanisms that is driving this. And the third piece of evidence is that there is local inflammation in the myofascial unit itself, and our colleague Dr. Jay Shah has shown that if you use microdialysis, you can actually see changes in a number of neuropeptides and catecholamines in the local unit. And the question is, why is this inflammation not being cleared? And so that's where we think that there might be impairment of this lymphatic drainage that sort of causes the stasis of the inflammation. So let me now show you a little bit of the data to back some of these claims up. So this is a paper from Dr. Shah's group where they used this microdialysis needle and looked at the local biochemical milieu in these trigger points, but also in a distal muscle. And they saw as you advance the needle, there is this alteration in these various neuroinflammatory peptides and catecholamines, including a change in the substance P, TNF-alpha, and so forth. And what is interesting is that right after you elicit a twitch response with this needle, you see a drop in these substances. So it means that there's some mechanism through which this is getting washed out or cleared. Again, this is just observations at this point. There is not a clear explanation for what might be going on, but this is one of the pieces of evidence that there is definitely an alteration in the biochemical milieu of these trigger points. And what is interesting is that there's a difference between what are called active trigger points or active myofascial unit, which is essentially a spontaneously painful tissue, versus latent, which is not spontaneously painful, but painful upon palpation. So in other words, it requires some kind of provocation to elicit the pain. So that's one data point. There's a series of imaging studies and that have been trying to generate some evidence of what might be going on. And this paper by Mazza et al. Sort of reviewed all the available studies until 2021. And so a lot of them were done using ultrasound. There were some using MRI. The infrared thermography studies were not very convincing. With the ultrasound studies, the main findings were that you could see these sort of focal regions of abnormalities on sort of anatomical B mode imaging. There's also some evidence of hyperechoic bands in the muscle. So I'm going to show some images to show what these look like. There are a couple of methods used to look at the mechanical properties of muscle, and I'm going to show exactly how these were done. But the evidence was that there were regions of heterogeneity in the muscle. So there were mechanical heterogeneous regions in the muscle. Some regions of the muscle were stiffer in affected versus non-affected individuals. And then there are alterations in the blood flow waveform that shows that there are some changes in the vascularity of that area. And on MRI, again, the MRI elastography data also back up the ultrasound data and shows these localized regions of stiffness in the muscle and in the taut bands that are palpated. And using T2 imaging, they showed a higher mean signal intensity near these regions of suspected trigger points. So that's sort of the evidence from the imaging study. So I'm going to show you a little bit of that evidence now. So the first is using ultrasound. So ultrasound is very attractive for using for musculoskeletal imaging because not only does it give you a very detailed sort of anatomical image of the architecture of the muscle, so you can very clearly see these fascicle bundles, you can see the connective tissue, and you can also see blood vessels inside the muscle. So this allows us to look at a number of different methods. Not only architectural and anatomical, but also potentially mechanical using this method called elastography. So that's one of the advantages. Ultrasound has a much better axial resolution in visualizing muscle architecture than MRI, for example. So that's another advantage of ultrasound. So ultrasound, of course, does not only show the architecture, but it can show mechanical properties. And there are a couple of ways in which that is done. One method is what is called a strain imaging, where you essentially press down with the ultrasound transducer. And as you're deforming the tissue, you can track the displacement, the local displacement of the tissue. And if there are regions that are stiffer, those regions will not deform as much compared to the surrounding tissue. So by tracking the deformation of the tissue, you can sort of try to infer what the local stiffness distribution is. So this is a fairly qualitative assessment. So you can look at these images and you can try to identify images that show local heterogeneity, but this is not necessarily a quantitative method. The second method that is often used is what is essentially based on a more percussive assessment. So this utilizes the radiation force of ultrasound to induce a local shear wave in the tissue. So ultrasound is mechanical energy. So if you increase the force with which you're pushing, you can actually induce this local shear wave. And so this video shows that shear wave propagating through the tissue. And as the shear wave is propagating, you can actually measure how fast it is moving. And the speed of that shear wave is a measure of the local mechanical property of the tissue. And you can look at both stiffness, but also viscosity. So it is a very quantitative way of locally evaluating tissue properties. So in terms of grayscale imaging, our group, as well as several other groups now, have shown that if you look at muscle with palpable trigger points, these muscles appear very heterogeneous on these kind of grayscale imaging images. One of the findings is that there are these hypoechoic regions that are localized, focal regions of hypoechogenicity. And that is one of the findings that we have reported and other groups have reported as well. The second finding is that there are these areas of thickened, sort of more echogenic, either perimysial tissue, but also the epimysial fascia are also potentially thickened. So groups have variously described these as hyperechoic bands or hyperechoic tissues within it. So you can see that as these regions of increased echogenicity within the muscle, and you can appreciate that in 3D as well. And some other groups have shown that there is a local thickening of sort of the epimysial fascia as well in individuals with neck pain compared to control groups, for example. And this, again, is consistent with this densification of the hyaluronan in these loose connective tissue. The other finding is that if you look at these images and try to quantify them using a texture analysis, we find that there is an alteration in the texture of these images. And so here is one example of a study that we did in our group, where we took these grayscale ultrasound images. We used entropy filtering as a way of looking at the local texture. And we found that there are these regions that appear to be of a different texture compared to the surrounding tissue. And if you compare a measure of the texture, so for example, the region with a texture parameter that's exceeding a particular threshold, you can see that there is a separation between a muscle that is palpably normal and asymptomatic compared to a symptomatic, spontaneously painful muscle. And we see that heterogeneity not only in the belly of the muscle, but also the regions surrounding this fascial border. And if you look at this in 3D, it appears that this heterogeneity has some underlying structure to it. So there are these bands of thickened perimysium that appear to be heterogeneous throughout the muscle. And so this is another sort of moment for us where we realize that the isolated trigger point that you can palpate is not necessarily the only thing that might be going on that's altered in the muscle. There's this sort of more heterogeneous alteration in the muscle itself that we should be starting to look at. The other method that we have used in our group is what we call vibration elastography. And the idea is, again, very similar to the concept of shear wave elastography, except that now instead of using the radiation force of ultrasound, we are using an external source of vibration. This makes these exams more technically challenging because now you have to hold this external vibration device next to the ultrasound probe. So it introduces some sources of heterogeneity in sort of collecting the data. But one of the things that we observe is that this allows us to really isolate these regions of heterogeneous findings in the muscle with more contrast than you can see on just regular grayscale imaging. So we can see these regions that are not vibrating and that are corresponding to this focal hypoechoic regions, but also we see these sort of bands that are not vibrating, and this could be consistent with this sort of thickened perimysium and epimysium that are sort of stuck together, if you will, and they're not moving as efficiently as it would in a normal tissue. So if you quantify these heterogeneous regions, we find that sort of the area of heterogeneity or a fraction of the area of heterogeneity is significantly different between symptomatic tissue and asymptomatic tissue. So again, it sort of emphasizes that normal tissue, normal muscle that is not symptomatic, does not have a palpable finding, appears to be very homogeneous with a uniform architecture on ultrasound. But these symptomatic tissues have various levels of heterogeneity, and we can identify this with reasonably good reproducibility. And another thing that we found is that if you actually treat this tissue using, let's say, dry needling, then the heterogeneity decreases over time, not only after a course of dry needling, but also at an 8-week time point. So then again, indicating that there are some ways in which we can perturb the tissue, let's say using dry needling, that seems to affect not only the muscle, but also the connective tissue surrounding it, so that this heterogeneity index seems to be dropping. But of course, the challenge is that there is a lot of heterogeneity in the variability in the findings at baseline. So this is the other part of what we are trying to make sense of, is why is there so much variability in the muscle tissue among these individuals. So if you track individuals with time, on average, they tend to decrease, but there's a lot of heterogeneity in the data itself. The last thing that I will mention is regarding blood flow. And so we have been using Doppler ultrasound to look at blood vessels in these affected muscle tissue. And one of the things that we observed is that there are these large blood vessels that are not supposed to be there from an anatomical perspective in the upper trapezius muscle. And they seem to be significantly larger than we expected in that local region. So we know about the normal sort of vasculature in the upper trapezius muscle, but these blood vessels seem to be occurring not where we had expected. And they tend to occur in these areas that we have identified as hypoechoic on these images. So these blood vessels they have large diameter, and if you look at the Doppler waveform, that means the change of blood velocity during the cardiac cycle, we can extract some sort of features from those blood velocity waveforms, and we found that, and at least in this one study, that there's a significant difference between, again, asymptomatic and symptomatic tissue. And the asymptomatic tissue seemed to have this sort of expected flow of a little bit of systolic flow and mostly quiet diastolic period, whereas in the latent and active muscles, we tend to see a sustained sort of diastolic flow, indicating a lower resistance vascular bed. And in these active trigger points, we sometimes saw this sort of reversal in the diastolic flow that indicated that there was some kind of mechanical obstruction that was preventing the flow from going forward in diastole, or some change in the local sort of pressure in the myofascial compartment that was preventing the flow from going forward at diastole. So the pressure in the compartment essentially exceeded the diastolic pressure. So this was an interesting finding. We are now trying to reproduce that in our new study. The other piece of evidence has been primarily shown in the low back, and this is from Dr. Langevin's group, who looked at the thoracolumbar fascia and tried to quantify sort of the sliding of the thoracolumbar fascia and look at the sort of shear strain in the various layers of the thoracolumbar fascia. And what they found was that in individuals with low back pain, there was a reduced shear. So in other words, again, consistent with the fact that there is this densification of the hyaluronan that causes the fascia to be more sort of viscous and less viscoelastic. And it does not allow this efficient gliding of the different layers of the fascia. So that finding was that in low back pain, there was a significantly lower shear compared to individuals without low back pain. And so this is an example from our study where if you look at the infraspinatus and the trapezius boundary, as a subject is trying to do an isometric contraction, you can see that this alteration in the gliding of the fascia. So in some individuals, there's this very nice shearing of the fascial layer where the muscles sort of glides past each other, whereas in others, they appear to be somewhat stuck and the fascia is not gliding as efficiently. So we are still working on quantifying this and trying to see if we can see a difference between different groups. We also use shear wave elastography in looking at the variance of shear speed as a function of the fiber angle of the muscle. So we know that the shear waves propagate faster along the fibers and they propagate slower across the fibers. So we are trying to utilize that as a potential biomarker to try to quantify whether there's an anisotropy, local anisotropy in the muscle. And so a preliminary study from our group showed that there was a significant difference in this anisotropy. We are now trying to do this in a more systematic way. So we hold the ultrasound probe with a collaborative robot, and the robot rotates the ultrasound probe in a very precise manner so we can quantify this shear anisotropy. This is ongoing work. In using MRI, there's been a couple of studies that showed the external vibration induces this changes in the shear speed. In the Todd bands, so there's a group at Mayo Clinic who showed that these Todd bands are visible on MRI. Other groups are now using T1 row mapping to look at the glycosamine glycans in the fascia layer. And the hypothesis is, again, that there's this increased densification of the hyaluronan, which is which would show up in the T1 row images. And so in this one study from NYU, they showed that if you look at this so alteration in the myofascial tissue of the forearm, and if there are regions that are tender, they tend to have this increased T1 row signal. And that goes away after a mechanical sort of perturbation with this sort of a force-based manipulation. And so just immediately following the force-based manipulation, there is an alteration in the T1 row signal. So just to quickly summarize, we see that there are a number of methods to understand what might be going on in the myofascial unit in terms of architecture, but also potentially the organization, functional properties in terms of the vascular physiology and mechanical properties. The MR elastography and T1 row could potentially be promising, but then there are a number of limitations. So a primary limitation is that it is very difficult to identify what is truly an abnormal group versus what is truly a normal group, because as many of you know, trigger points are ubiquitous, even in asymptomatic individuals. So really trying to figure out what is a group that needs to be studied in terms of abnormality in their myofascial unit versus what is normal physiological variation. That is a tricky challenge, and we're still trying to better understand now how we are going to be able to do that. So I just wanted to highlight a couple of other sort of opportunities coming up. So there's other modalities that are still not being investigated, but these are available. So the contrast enhanced ultrasound can give us a good understanding of perfusion. There are methods now that can provide very detailed microvascular flow maps of tissues. So that could be an opportunity to look at altered vascular physiology. Photoacoustic imaging, where light is sent in the tissue, and as the light is absorbed, it generates a little sound wave. And that can be used for looking at sort of optical properties, including things like oxygen saturation and other sort of molecular mechanisms in the tissue in human subjects. So this is, again, a very exciting advance that is still being investigated. Of course, ultrasound technology is getting better. So there are ultra high frequency ultrasound probes that are commercially available now that provide much more detail in terms of what we might be able to see. So this, again, has not fully been investigated. In our group, we are now developing ultrasound sensors that are wearable. So we are no longer limited to sort of a handheld ultrasound system. We can actually put these little ultrasound patches on individuals while they are doing a dynamic task and look at how the alteration and the function might be happening during a dynamic task. So this is, again, an opportunity to investigate sort of more dynamic function, including things like fascial gliding and so forth. So this is all potential future work. So I wanted to close by talking a little bit about our ongoing HEAL study. So this is, we're focused on neck and shoulder pain. Again, we are trying to look at multiple modalities to investigate the dysfunction in the myofascial unit. So we're using shear wave elastography as a function of angle. We're looking at this gliding of the fascia. We're looking at blood flow, but we're also using other methods like bioimpedance spectroscopy to look for sort of local changes in intracellular and extracellular fluid. We are also using high-density EMG to look at local changes in the motor unit excitability. So we are hoping to develop a quantitative biomarker that can tell us a difference between different types of phenotypes in the myofascial unit in individuals with pain and individuals without spontaneous pain. So this is ongoing work. We are in our second year of the study. And so this is an example of the types of data that we are collecting, including bioimpedance spectroscopy, a very detailed ultrasound with these robotic manipulation of the ultrasound transducer, so to improve reproducibility, as well as more dynamic studies while an individual is performing isometric contraction, where we're looking at gliding and the EMG. So I'm going to stop by acknowledging all my collaborators who have been working on this project for a long time, and our funding from the National Institutes of Health. And I'm happy to take questions.
Great, thank you, Siddhartha. Great talk. Indeed, the floor is open to questions if anyone wants to jump in.
I had a question. Just wondering, so I know that this group is full of a lot of different people, and I know part of the reason why this, you know, group exists is to kind of promote diversity within the field. And so I'm just wondering why that wasn't really accounted for in the gender data collections. I noticed there was only male or female, and then also beyond that, are there even trans patients included? And if so, how are they being categorized? I know I was really excited that your studies are taking into account the social kind of factors, because I think that's very important when talking about chronic pain. I think we can all agree. And if we are actively just not even acknowledging trans people exist in our studies, then that kind of goes against, you know,
Yeah.
mission and science. So I was just wondering,
No,
you had anything to share about that?
thank you so much for that question. So I will say that we are actually not just collecting data on just a binary gender. We are allowing individuals to sort of self-select gender in our ongoing study. Historically, in the past, we did not. And as you know, this is one of the challenges. And I think your point is extremely well made, and that we really need to better understand sort of the experience of chronic pain in different populations and really understand what might be going on. So one of the things that we are doing going forward is, it's one thing to look at these biomarkers in the tissue, but clinically, this diagnosis is not usually just based on just one clinical examination or one physical examination. It is usually a dialogue between the patient and the clinician, and that dialogue is a long process. So, in a community-based clinic setting, that is a problem solving between the patient and the clinician. And typically in research studies, that nuance is completely lost, right? And I think you are trying to allude to that point, is that if you use a questionnaire where the researchers have pre-selected what questions that they are interested in, that completely misses all that nuance of that complex lived experience. So we are very aware of this problem, especially in chronic pain. So we have actually convened a community advisory board of the patients with lived experience, of the physicians in the community, and they're part of our advisory board, and they are trying to help us design better ways of sort of collecting this rich experience of chronic pain. So please stay tuned. I know our, this is, we are all understanding slowly that this is a very complex problem that might affect different people differently. So we are aware of this. Have we solved the problem? No, there's a lot of work to be done. Thank you for that question.
Yeah, and thank you also. This was really, really beautiful, beautiful work y'all are doing. So thank you for that.
I am wondering, is there, have you looked at all at kind of momentary or state-based changes in these various properties, even played with it? So for example, if someone is anxious and tense, which could be a result of their pain or the biopsychosocial processes, have you looked at how that is there indications of to what extent that impacts these different biomarkers?
That's again an excellent question. We have been thinking about this a little bit in our group. So one of the things that we do in our current study is that we send patients home with an app, and they sort of track their pain experience and also their activity on a daily basis. Twice a day. So we have been doing this for 90 days in our current patients. So they give us a very detailed information about their pain experience on a daily basis. And what we find is not surprising, but it sort of shows us the complexity of this. So the pain experience is obviously not necessarily representative of what their baseline findings were when they came into the lab for the clinical exam. So in some patients, there's this very episodic nature of pain. So Even on a daily basis, there is a lot of fluctuation. For others, there's a more slow episodic nature. So the pain increases and stays high for a long period of time, and then somehow resolves and then returns back. And so the challenge is, it's very hard for us to design a study where we are systematically looking at this detailed imaging on this fine granularity. But we are hoping that in the future, we should be able to do that with at least a couple of these biomarkers. So for now, we only have sort of patient self-reports that tells us about this sort of temporal changes in pain, but we don't quite have sort of a detailed biomarker data. We do look at these patients for at least a couple of different time points. So we have some data on different time points, but that may not be linked to what their pain experience might be. So they may come in to us on a day where they're feeling completely pain-free, and we look at their tissue, and that doesn't mean that the next day they're not going to be in very high pain. So this is a very complicated challenge with studying chronic pain.
Thank you.
Harry, do you want to jump in? I think you were up next.
Sure. First of all, thank you so much for the work that you're doing in your group in this area. It's very important, and we appreciate that. And question, do you have a hypothesis on why the trigger point or tender spot itself would be more hypogenic versus hyper?
It's a very good question. And, we have been puzzling over this for a long time, and I don't think we have a satisfactory answer yet. So there are a couple of different things that we can postulate, right? So we know why tissues appear to have different echogenicities on ultrasound. So there's some underlying sort of theory for the physics of ultrasound and how it interacts with tissues that leads to changes in echogenicity. And one of that is that, if there are alterations in sort of the tissue composition, so for example, if there is one tissue that has an increased fluid content, that will appear to have a different echogenicity compared to the surrounding. The other is architecture. So if there's alterations in the microarchitecture of the tissue, so that could also lead to changes in echogenic properties. So when we first started seeing these things, we thought that it could probably may have something to do with fluid, but increasingly we realized that if you look at the tissue at different angles, this is not a pocket of fluid in the tissue. That we can rule that out. It is. I don't think it is an isolated pocket of fluid. Like for example, if you have a local injury and there is a local pocket of fluid in that tissue immediately, that appears very different from what we are finding. So there's some changes in the tissue composition and architecture, but it is not entirely clear what that is. I don't have a satisfactory answer to your question yet, but we are still puzzling over it.
Well, thank you. And do you think is there a possible link between the changes in the blood vessel structure? And also, is that like neovasculogenesis in the area with like collateral branching, or do you think that it's just vasodilatation?
So I think that it's a little bit of both. So I think there is, we were very surprised to see these blood vessels in some of these patients, especially in the areas that we were seeing it, because we know what the normal vascular anatomy, the trapezius is. So these blood vessels were clearly not the main blood vessels that we had expected in that area. So, essentially there were either neovascular neovasculogenesis or potentially small blood vessels that have now become dilated. So it could be both.
And do you think that might be related to anoxia in the area that the body's responding?
So there's been some evidence that there's local hypoxia in the tissue. And this was one study that advanced a needle with a oxygen sensor, the tip of it, and that showed that there was a local sort of a region of hyperoxia and then a region of hypoxia inside. That study, unfortunately, we have not had a chance to replicate that yet because technically these are very difficult to do. But there are other groups that are developing these sort of needle-based sensors, and so I'm hoping that we will have better answers for you soon. We are also very excited about the use of photoacoustic imaging to try to answer this question, because photoacoustic imaging does give us a map of local oxygen saturation. So I'm hoping that with photoacoustic imaging, we might be able to answer this question.
Natalie, I saw your hand was up.
Yeah, I was wondering, in regards to the innervation to these myofascial units, do we know anything about the specific types of like nerves that are innervating this, like nociceptors, proprioceptors, and whatnot?
Yeah, and the answer is both, right? So there's both. So the muscle spindle apparatus is actually located in the fascia itself, right? So the capsule is continuous with the perimysial fascia. So if there are alterations in the sort of the gliding mechanics of the fascia, that's going to impact the proprioception. So that's that much I think we know. In terms of the nerve ending, so there's a lot of not a lot, a couple of studies that have really looked into this, primarily from Carla Stecco's group and Antonio Stecco's group. And I'm happy to share those papers with you that show that, sort of the innervation of the fascia. But this is an active area of research. These are relatively recent findings.
Thank you.
Okay, we are almost at time, but if there are any final questions. Okay, thank you all for attending today. Siddhartha, thanks for your talk. It's great. We really appreciate your time today, and best wishes on your continued research.
Thank you so much, and thank you all for your attention and your excellent questions. And thank you for inviting me. Yeah, this was great.